Hi, thank you for joining me today. My name is Mikaela Fetter, and today I will be discussing with you the influence of predation on the stress response. Before beginning my talk, I would invite you to scan the QR code at the bottom of your screen shown here. This will take you to a website where you can view the figures larger and learn more information by viewing my references and our lab website. We know that predators have profound effects on their ecosystems. They can do this by affecting the abundance of prey, which can affect the biodiversity, but they also can affect the physiology of the organism through the stress response, which can affect other systems in the body as well, which I'm going to talk to you more about today. What we don't know is how this effect is affecting the systems long term. My study will seek to better understand the evolution of the stress response due to variation and predation. Our lab studies the Trinidadian killifish, which are located in high predation sites and killifish only sites, which makes them an excellent model to study the question at hand. These populations differ in life history strategies and morphology. We predict that killifish in high predation environments will have higher baseline cortisol levels as predators can increase HPI activity. The HPI is the hypothalamic pituitary adrenal axis. Stress is received by the brain, which causes uh, a cascade effect to then release cortisol. Cortisol is the stress hormone, and this is short term really good because it increases the likelihood of survival by mobilizing energy through the increase of glucose. However, long term, this can have some detrimental effects as it can modulate energy from the immune system and has been shown to be linked to decreased growth and reproductive success. My research is twofold. First, I plan to examine the differences between the two populations of the hybridation fish and the killifish only populations to establish baseline levels in wild populations. I plan to extract the cortisol and quantify it with an enzyme-linked immunosorbent assay, an ELISA. Second, I plan to develop a non-invasive water detection method to quantify cortisol in second-generation lab-brood fish. Will work as fish leach their hormones via their gills. This is also really exciting because then rather than just measuring at one point in time, we can look at how cortisol is varying across time for individuals, as shown in the graph here. So we could quantify a baseline level, look at inducing a stressor like a predation cue, and then see how it changes over time. I do have preliminary data here, which is showing that high predation fish overall have lower cortisol levels than killifish only predation or killifish only populations in F2 generations, which is the evolved population. This is exciting because one, it has validated my methods. Two, we see differences in the populations. And three, that this is really interesting as it's different from what we predicted. In light of this adaptive response that we see and thinking about going forward as some other responses, I would also like to think about the cortisol trade-off hypothesis and the fact that there is life history trade-off in the protective and damaging effects of glucocorticoids. As cortisol increases, you're more likely to survival, going back to think about that fight or flight response. However, there is a negative relationship with reproduction. So I would like to further quantify reproductive hormones in these species, in these two populations, to further think about the adaptive response with environmental stressors. I thank you so much for your attention today, and I look forward to answering any questions and continuing to conduct this research.